Our project is about determination of polychlorinated biphenyl congen, otherwise known as PCB, in water by liquid-to-liquid microextraction and gas chromatography. PCBs are toxic and carcinogenic organic pollutants. Altogether, there are 209 congeners, which are classified into 3 main classes based upon chlorine orientation. They can be transported over long distance through soil and sediments and accumulate in food chains. They also pose dangers to human health. The report will determine 10 representative PCBs in rainwater and bottled mineral water. And our analytical methods used will be liquid-to-liquid microextraction coupled with capillary gas chromatography and electron capture detection.
As mentioned previously, the technique we are focusing on is gas chromatography. The diagram shows a general schematic representation of the GC. For the experiment, the carrier gas, helium, is injected at 1 ml per minute into the oven. Oven conditions are programmed as such. Specifications of the capillary column are as such. To illustrate further what happens in the oven, the sample is introduced into the oven and is carried to the detector. The sample is then processed by an integrated detector called electron capture, in which the detector response is reflected on a chromatogram. As the different compounds pass through the column and interact with it, they will be eluted according to their interactions with the stationary phase. The analyte which has weaker interaction will be eluted first, followed by the stronger ones.
Data collection. This is the representative chromatogram of rainwater with spiking level of 50 nanogram per liter. The 10 different PCBs are eluted out at different times. Sources state that the retention time is based on the number of chlorine atoms. This corresponds to the data achieved as seen from the structure of PCB9 and PCB209. Results from triplicate assay before and after spiking at 150 nanogram per liter are tabulated in the form of percentage recovery plus minus RSD. Percentage recovery refers to the amount of analyte recovered from the 2 water sample. RSD refers to the relative standard deviation. These are the results obtained from the table. Rainwater gives us both the minimum recovery of 80% and highest RSD of 7.8%. Reasons for this could be due to low concentration affected by the negative matrix effect. Possible reasons also include formation of complexes that hinders with the recovery.
The first point for criticism would be that there are no variation of conditions for GC analysis. The conditions mentioned earlier are the only conditions stated in the paper. However, another source states that GC analysis conditions such as temperature of the column, gas flow rate and gas carrier are vital for better resolution of the peaks. Next would be that there is no LOQ value reported in the paper, but only a LOD value of 5 to 15 nanogram per milliliter is reported. However, as we know that LOQ is actually more accurate than LOD, the paper could have reported both LOD and LOQ values similar to the source shown. Next would be that there is no comparison between different solvent used for extraction, as only hexane is being used due to the fact that PCB is non-polar. However, another source which did a comparison between solvents for extraction reported that toluene gives the highest average recovery. Our last part would be that only spiking levels of 50 and 100 nanogram per liter was done. In my opinion, I think that this is purely random, so as to get the experiment started. This also could be an area for other researchers to extend the research using different spiking levels.
It is concluded that there are 10 types of PCB present in the water sample analyzed with good percentage recovery and low standard deviations. The technique used for this paper is the liquid-liquid microextraction coupled by capillary gas chromatography with electron capturing detector. Our focus of this paper is the capillary gas chromatography. This technique is chosen as compared to conventional liquid-liquid or solid-phase extraction due to advantages such as low cost of extracting apparatus and shorter analysis time. It is concluded that there are 10 types of PCB present in the water sample analyzed with good percentage recovery and low standard deviations. Our group would like to point out some critical areas of improvement, which include implementing variations when conducting GC, for example, changing carrier gas and carrier gas flow rate, and include more variations of solvent use.